Welcome to Simon's Bench Talk, the podcast where we bring the most exciting science right now from the field straight to you. I'm your host, Samantha, and today we're going deep into a topic that is very complex and fascinating, the immune system. We are not going to talk about the immune system as a whole, but on a single cell level, so about individual cells. My guest today is Sander Wuyts. He's a co-founder and CEO of ImmuneWatch. And just to give you a little bit of background on ImmuneWatch, it's a biotech company located in Belgium. And they are doing something genuinely remarkable. They are putting a name and a function and a meaning onto the billions of unique immune cells within our body. So welcome, Sander.
Hi, Samantha. Thanks for having me. Happy to be here.
So before we talk a bit more about ImmuneWatch, I want to set the scene for the listeners. If you had to explain immune repertoire profiling at single cell resolution over a beer, how would you put that?
First, most important, what kind of beer? Is it a Belgian beer or a German beer?
Of course, Belgium.
Okay, that's the answer I needed to hear. No, thanks for that question. Immune repertoire profiling, I always think it as like a huge potential. If you think about your immune system, it's actually a collection of cells floating around in your body, trying to protect you from everything. And it contains a lot of information. It contains information about the past, what kind of infection and vaccines you might have had in the past, what kind of disease you're having now, because if you're having a disease, if you're infected with something or have a cancer, there's a great chance that your immune system is fighting it. So this is something you can immediately notice from your immune system. But it also has like a future predictive power. It can be used to predict how people actually will respond to kind of different therapies. So if I think about immune repertoire profiling, it's about characterizing all of your potential things you had in the past and how you're currently battling diseases. I was actually trained as a microbiologist. I also see a huge parallel with the microbiome field as well. So in gut research, we actually used sequencing as well to characterize what kind of bacteria or yeast or fungi are available in your guts. And I see a huge parallel with the immune profiling. So we use similar technologies to characterize what immune cells you have compared to what bacteria you might have in your gut.
So the gap that the field had, and correct me if I'm wrong, was basically that you could sequence the receptors, the TCRs, and you could see them being up or down regulated, but you had no idea what they were actually reacting to.
Yeah, that's one of the key things we're working on at ImmuneWatch. So if you compare all of this, like with microbiome sequencing, so where I actually did most of my PhD postdoc work in, if you would sequence a gut sample, a stool sample, the first thing we do is we will extract DNA, RNA sequences, and we would then try to map these to what bacteria is exactly in there. And this bacterium would then tell a whole lot about maybe your diet and how the healthy state of your gut. With immune repertoire sequencing, this is not the case yet, because if we just take a blood sample, for example, PBMC sample, and then do T-cell profiling on it, we get a whole bunch of DNA or RNA sequences that are mapping to these T-cell receptors. But doing this translational step into what the exact function is a very hard one, because we just see T-cell sequences going up and down, for example, after an infection, for example, after a vaccine. But what exactly these T cells are reacting to, that is a huge gap in the field. And this is also something where we are working hard at ImmuneWatch. This is actually the kind of functional layer that we at ImmuneWatch are closing and have closed already for quite a substantial amount of applications. So what we do is we take these T cell sequences and use machine learning and database matching to figure out what these T cell exactly binds to or recognizes, whether it's a SARS-CoV-2 specific T cell or a melanoma specific T cell. We are this kind of intellectual layer that we put on T cell sequencing data to extract more meaningful insights from immune repertoire data.
Right. So let's talk a bit more about ImmuneWatch. You already mentioned your background is actually in microbiome research and not immunology. So how does someone who spent their PhD studying bacteria end up founding an immunology company?
That also has been an interesting story. And over the past years, I've actually been introducing myself on kind of different ways. I always felt like a scientist, but also a molecular biologist, then a bioinformatician, then a biologist, and now maybe half of an immunologist, because immunology is really a hard field. I think the key link is the molecular biology, though. My PhD postdoc was all about finding signals in next generation sequencing data sets. So DNA sequencing data sets from microbes, from human samples, from gut samples, from vaginal samples. When I was doing my PhD, I did a lot of bioinformatics analysis for the lab that I was working in. And I was reaching out within the University of Antwerp, where I did my PhD, finding other bioinformaticians, other people that not doing exactly the same kind of work that I was doing, but also analyzing next generation sequencing data sets. And this is how I actually met my co-founders, Peter and Chris, who were already doing at that time, together with Ben, a lot of T-cell sequencing. And I immediately saw the methodological overlap, like 16S sequencing for microbes and T-cell sequencing for immune cells. We both use very similar computational approaches. But you can argue that this is for a lot of these omics data sets these days. But the kind of analysis techniques that we were using bioinformatically are super similar. And at that time, I was also thinking like, hey, this is an interesting emerging field, this immune repertoire profiling. And compared to the field that I was working in, microbiome field, I kind of felt like the T-cell sequencing field was running like maybe 5 to 10 years behind the microbiome field in terms of maturity. I felt that microbiome sequencing was applied almost everywhere where we could find microbes, while for T-cell sequencing, it was still emerging. This is also an interesting opportunity, right? Because you figure out the impact that transitioning from regular sequencing or regular cloning techniques into next generation sequencing methodologies or single cell sequencing methodologies. If you can spot that shift, then as soon as people switch to the new generation technology, a lot more data will be processed, and you need also quite some new computational methodologies to then be applied to crunch all of this data. So for me, the signal was clean. I saw an opportunity there. And I also really much believe that the immune repertoire is a much more actionable layer compared to microbiome sequencing. Because if you are reading out a microbiome, you just screen what bacteria are there. While if you are actually reading out the immune system, you can really see immediately how a body is reacting to a vaccine, to a therapy, or to an infection. I think that's a much clearer and faster actionable insight compared to a shift in the composition of microbes.
Yeah, I agree. I mean, I find it very interesting seeing that you had expertise in one field and you kind of applied it or transferred it to another field to accelerate that one. I wonder, was there a specific moment where you thought, okay, this has to become a company, we have to do this?
So I am also very grateful to my co-founders, of course, because when I just started out as a CEO, I barely had any immunology background. And so my co-founders who are rooted in immunology, they really helped me out in seeing all of these things and helped me figure out also what kind of applications are now the most interesting to pursue. So it's definitely not a one man's job. It's a team work and a team effort. And nevertheless, I think we all felt at that time we were developing machine learning algorithms for making immune repertoires much more actionable and more insightful. And we knew it was also after the COVID crisis. So we knew the importance of what it is to track immune system responses after vaccines. We had the emergent mRNA vaccines, which are also used in oncology. So this all gave a boost to the whole vaccinology field, or even the whole cell therapy field as a whole. So I think this was the pivotal point where it was interesting to say, hey, this is an academic tool. This was interesting performance. But if you really want to take it to the next level and want to serve biotech and pharma companies, it might be the time to start developing and working on a better database for machine learning and a better tool that's easy to use in pharma and biotech. So that's one thing. I think we had a lot of confidence in that at that time, there would be a huge market adoption of immune repertoire sequencing, and that we just also need more computational tools to power that all of the data that has been generated to really extract meaningful insights out of that. So I think, yeah, everything came together at that time, and that's why we decided to make the jump out of the university into a company.
You just mentioned that ImmuneWatch was more or less founded around the time where COVID emerged. So, where does it matter clinically what you do? Where does this come into place there?
Yeah, initial focus of papers from my co-founders were also mostly in infectious disease spaces. So we were also thinking that basically it could be used for T-cell sequencing, and then our analysis tool could be used for monitoring vaccine responses in infectious diseases. But then we also learned that infectious diseases and the way they monitor vaccine responses is maybe they have a traditional way of doing that, wet lab assays, ELISpots, etc., flow cytometry. And these companies working that space were maybe not also the first movers to move to new technology, to immune repertoire sequencing as a way to monitor how people respond. So this is something we actually kind of learned relatively fast in the first year of founding the company. And I think this is also always a challenge. If you spin something out from an academic institute, you have assumptions about the market, and these assumptions really need to be tested. And so while we were first like, hey, infectious disease companies will be extremely interested in what we're doing, this proved to be wrong. And we kind of pivoted there into oncology, because that's where you notice that in oncology, where you develop new kind of vaccines, therapeutic vaccines instead of the protective vaccines, or other kind of cell therapies in oncology, they need to adopt all new ways of monitoring how their patients respond actually.
Right. Can you walk us a bit through the process there? What is the impact of your technology on that?
Yeah, so currently the sweet spot at ImmuneWatch is like phase one clinical trials in oncology, or very early in patient trials with new oncology products like personalized cancer vaccines. Because is these trials, they are of course made to test safety, but a lot of these companies also want to see early signals of efficacy, and this is where we actually from a research side try to help them and squeeze as much possible research information out of this. So the sweet spot is definitely oncology now for us.
Mhm.
Regarding personalized vaccines, this is always kind of a dream where we want to go through with companies, but it's also a hard field, because what we work on is machine learning, and we have a good training database, meaning that if you give us a T-cell repertoire, we can match it with these TCRs with antigens or epitopes stored in our database. And so these are usually targets like tumor-associated antigens that are very well described targets. The algorithm works very, very well in that use case. But for a personalized cancer vaccine, by definition, you will work on some neoantigen or a neoepitope that's highly unique to this patient. And that comes with an additional challenge, because these data are by definition not stored in our database. So it's a challenging field, and we still hope to be at the forefront of that, and we are at the forefront. But we have been working on quite some cool use cases. For example, a personalized cancer vaccine encoding 12 to 20 epitopes that
Mhm.
are designed from specifically using the tumor material of the patient. And then the question was like, hey, we have a couple of these epitopes in the vaccine. Can you now use your technology, T-cell sequencing, to fish out which of these epitopes now actually caused an immune response? Basically answering the question, what part of the vaccine worked and what part not? And this creates a very cool feedback loop, because if you're able to do that, then you can actually re-guide your vaccine design for the next patient or for the next personalized vaccine. And so this is where we helped in a couple of use cases. We're not completely there yet to apply it from any use case, where we could really fish out, oh, these two epitopes, we clearly see a T-cell response there using T-cell sequencing and the algorithm, while for the other epitopes, not. And that's really the kind of feedback loop that would guide vaccine design for the next round.
Yeah, that sounds really cool. It sounds like it's really driving forward the research in that field. Let's zoom out a little bit here and talk about something that's been on the news. Hantavirus has resurfaced as a concern. So, when you see an emerging viral threat like this, what is the first immunological question that goes through your head?
I'm extremely biased, of course, because I'm always on the T cell side of things. And in immunology, you always have these famous antibodies or B cell and T cell kind of camaraderie, I would call it. So my first thought is always, do we know what are the key antigens, and are they also causing a T cell response? Because from COVID and other infectious diseases, we learned that antibody responses are extremely important. They're a very good first line of defense to prevent infection, for example. But their effectiveness can wait or fade as the virus mutates into new variants. And in contrast to that, if you look at T cells, they can provide a more long-lasting, a more broad protection compared to antibodies. And they are really the critical safety net to prevent hospitalization and deaths when antibodies actually fail. So
Right.
again, I'm biased, but I would scout for antigens that also lead to a protective T cell response next to antibodies. And I think with like SARS-CoV-2, it was clear that we had to go for the spike protein. The antigen was relatively easy found, and it unlocked fast vaccine design. I'm no expert on hantaviruses, so please call me out on LinkedIn or email later on if you do know antigen targets. But my first approach would always be run computational tools and wet lab assays to try and find immunogenic peptides or antigens that also cause T cell responses. So you can use multiple computational tools for that, or really go into the wet lab and then test them out. Next step would then be, for me, is design a proof of concept vaccine, either mRNA, peptide, or any kind of vector, and try to see whether you can go preclinical and clinical into causing really a T cell response. And this is then where we actually could step in then later on, measuring whether there is a T cell response using T cell repertoire profiling, single cell level, or bulk profiling level, and then try to match these TCRs whether they are indeed epitope or antigen specific. So I would say we have quite some computational pipeline as well to already improve these steps. But the proof is in the pudding, and if there's a new virus X coming in, I think we need to deploy them as fast as we can, and people are working on this, like CP is doing that. We have much more tools available, I think, but they remain to be battle tested with speed, I think.
Right. Yeah, I think that's a really clear framework, and it shows clearly where tools like ImmuneWatch can slot into pandemic response pipelines. So, yeah, basically, for any novel pathogen where we would start from scratch in immunology, that is how you would do it, right?
Yes, new emergent pathogens, but of course, there's already a whole body of knowledge for maybe related pathogens. So indeed, for SARS-CoV-2, we had SARS-CoV-1 and MERS, where we could also use that knowledge. I would always also consult experts in the field as well, and not only rely on what the computer tells you to do.
Of course.
There's a lot of knowledge out there in virologists all across the world, so I would also go and look at them and see what these experts have to say on antigen level, and then next to that also run the computational pipelines.
Right. Okay, let's talk about what actually is exciting you right now. I mean, you go to conferences and you see what's coming. What's had you thinking recently? This is going to be a change in the field.
I think AI is definitely one of them, but I'm also excited about new sequencing technologies. And what I noticed is the in situ sequencing or the spatial kind of technologies that are also moving into TCR sequencing and T cell sequencing. So I know it's probably very niche, but for me, it's an emerging field. It would be really, really cool if you can actually take some samples, tissue samples, and then next to doing the RNA sequencing on these samples, also pinpoint the exact TCR sequences that are, for example, in a tumor sample or in tissue sample. And if you can then actually sequence these specifically very locally and then overlay our technology onto it and find the specificity, so really looking at a microbial, at a slide and figuring out, oh, okay, these T cells locally, these are specific to these antigens and these specific to these antigens, that really excites me. So I'm hoping to see some applications of that in the coming years, actually.
Right. You mentioned AI. I want to pick up that topic again. So, AI and machine learning, there's a lot of noise right now and a lot of hype. Is that justified, or are there also limitations that you see?
I think most of the AI hype also comes from the LLMs, and I think they're doing great things. I think for my own productive workflows, LLMs are really doing some heavy lifting that I had to work a day on, they can do in a couple of hours. So for me, it's really time saving and real productivity gains. But for like highly specialized niche annotation tasks, like predicting T cell epitope specificity, I think general LLMs are just not there. We really, really need domain expertise to catch good algorithms in this space. So I think indeed there's a lot of things going on in the LLM world, but also maybe AlphaFold worlds, 3D structure worlds. There's a lot of things going on. But the really niche things, I really believe that you sometimes need to develop in biology very niche models that have a lot of domain expertise added to this. And we see this in a lot of deep tech companies. They actually focus on one specific task and try to be the best at solving that task. I think where this kind of specialization is really important to make the difference, because we can talk about foundation models that are trained to capture everything, but biology is inherently noisy. The data that we're generating is still noisy. It's completely different to like training an algorithm, an LLM on text or speech data, because the data in biology is just not at that level yet. So that's why I'm a firm believer of building and focusing on models that are very good in niche tasks, and maybe in 5 to 10 years we can talk about something else, because then the AI wave might have catched up there. But for biology, I think it's a key thing to generate more data and more specific data for your use cases before we can talk about generalization.
Okay, so kind of wrapping all of that up, what do you think? Where does all of this go? Fast forward 10 years from now, what do you think? What does immune monitoring look like in the clinic?
I think T cell sequencing, or I hope that T cell sequencing is going to be a very routine part of all of this. I think it's a really powerful tool. Like I actually started the podcast here, as I think an immune repertoire can give a lot of information about the past, what infection, what vaccines, a kind of immunological passport actually, or immune passport that's really stored in your immune system. But it also has a lot of information about the present, so a diagnostic tool. So really is showing how people are responding now, really in the clinic, like figuring out, hey, you have multiple time points and the immune system has evolved over this time. What actually is it trying to combat? A single blood sample will actually be used to then classify multiple diseases at the same time just by looking at your T cell or maybe even B cell repertoire. So I'm really hoping to see more diagnostic and prognostic kind of applications out of T cell repertoire sequencing. And I think we need the kind of tools to unlock that translational layer as well. And I think that should be feasible in the next coming 10 years.
I love that framing. I mean, we at Synchron, we've also tried to move single cell sequencing more into the clinic, moving towards precision medicine 2.0. So it's really nice to see how that can also connect with immune repertoire profiling, and how both of these can move into the clinic and really, hopefully in the future, make a change for the patients and for how we treat patients.
Exactly. I think the tools that are being built now are already a good foundation, and they're extremely useful already in research questions of samples coming from the clinic. But the field also needs to move beyond that. But we're now putting that infrastructure in place, I think, for the next coming 10 years to really grow that into routine clinic as well.
Sander, this has been a wonderful and fascinating conversation. It was great to hear about the founding story of ImmuneWatch. It was really interesting hearing about the personalized cancer vaccines, and hearing your take on how we would meet emerging viral threats or new pathological situations. So it was really great hearing your insights into what is coming up in the field, what are the new and exciting things. So thank you for sharing your insights there, and thank you so much for being on Simon's Bench Talk.
Thank you for having me. I always get excited about talking about T cells, T cell repertoire sequencing, and also about my career or where the field is going. So it was a pleasureful conversation. Thanks for having me.
Yeah, I could definitely hear that. So, for everyone listening, if today's episode got you thinking about where your research meets immune profiling at single cell resolution, that's exactly the conversation we want to keep having. So, yeah, links to ImmuneWatch and Singleron you can find in the show notes. And thanks for listening.